So good afternoon. Thank you very much for this kind introduction. And first of all, I would like to deeply thank the MMF for this kind invitation to be here today. It's really a huge pleasure to attend such a great conference. So my name is Pedro. I'm currently a postdoc researcher in Professor Jesus Banali's group. And today I will talk a little bit about our AMMF-supported work on the role of the transcription factor KLF5 in the etiopathogenesis of cholangiocarcinoma. So Kruppel-like factors, or KLFs, are a family of transcription factors that include 17 members that are key regulators of tissue homeostasis, organogenesis, metabolism, also regulating cell differentiation and proliferation in all the human body. All of them regulates homeostasis in all the organs, and it's not surprisingly that these KLFs are all dysregulated in human disorders. And cancer is not an exception. It is widely known that KLFs strongly participate in the carcinogenic process, being involved in the regulation of cell proliferation, survival, and cell death, also regulating angiogenesis, EMT, cell migration, and metastasis. One of the major roles of KLFs is also being involved in the regulation of cell cycle, as they regulate the activity and expression of different cyclins and CDK, which are proteins that regulate the progression of cell cycle. And as you can see here, there are several KLFs that may act in distinct parts of the cell cycle. And among them, we can see that KLF5 appears in different steps of the cell cycle. So what is KLF5? It's one of the members of the KLF transcription factor family that is highly expressed in the gastrointestinal tract with the highest expression in the intestinal epithelia, in the colon, and in the stomach. Through the regulation of all these targets, it is known to regulate cell proliferation, apoptosis, differentiation, and migration. Still, it is important to know that the role and the function of KLF5 is dependent on the cell in which KLF5 is being expressed, since KLF5 may undergo different post-translational modifications that differentially regulate its activity. We know that KLF5 is dysregulated in different types of cancers, but we know that this dysregulation is cancer dependent. And KLF5 may act as a tumor suppressor or as an oncogene. For example, in esophageal squamous cell carcinomas and in prostate cancer, KLF5 is known to be decreased, acting as a tumor suppressor, while in, for example, colon cancer, breast cancer, bladder cancer, lung cancer, and other types of cancer, such as pancreatic cancer, KLF5 is markedly upregulated, acting as an oncogene. But what happened in cholangiocarcinoma? Up to now, there is no description on the role of KLF5 during cholangiocarcinogenesis. Therefore, we hypothesize that KLF5 may contribute to the development and progression of cholangiocarcinoma, potentially representing a target for therapy. In order to address this hypothesis, we developed distinct aims, and our first aim was to analyze the expression levels of KLF5 in human cholangiocarcinoma tumors. To do so, we evaluated the expression of KLF5 in 5 international independent cohorts of patients in which we observed that KLF5 is markedly upregulated in human CCA tumors, as we can see here in red, when compared with either surrounding liver and also with normal bile ducts. Importantly, when we stratified the patients from the Copenhagen cohort according to the most common genetic mutations found in these patients, such as the IDH1 mutations, KRAS, and TP53 mutations, we observed that KLF5 is equally upregulated in all of these patients, and when compared with surrounding liver and with patients with undetermined mutations, which led us to conclude that the upregulation of KLF5 may be a general event during cholangiocarcinogenesis. Next, we decided to evaluate if KLF5 is related with prognosis. To do so, we stratified the patients in these cohorts according to the expression levels of KLF5 in higher and lower expression levels, here represented in red and blue. As we can see here in these 3 cohorts, we observed that patients who displayed higher KLF5 expression levels displayed a worse overall survival, and importantly, the patients that presented a positive lymph node invasion displayed a highest KLF5 expression. With these results, we may conclude that KLF5 is actually also a prognostic factor in cholangiocarcinoma, being related with worse overall survival and with lymph node invasion, potentially related with metastases. We also evaluated the expression levels of KLF5 in human cholangiocarcinoma cells in culture when compared with normal human cholangiocytes. And when we evaluated the expression in these 8 human CCA cell lines compared to the 4 normal human cholangiocytes in culture, we observed that the mRNA levels of KLF5 were markedly upregulated in all the CCA cell lines evaluated when compared with the 4 normal human cholangiocytes. And this happened not only at the mRNA level, but also when we measured the protein levels of KLF5 in 3 cholangiocarcinoma cell lines, both at the total protein extracts and also in the nucleus, we were able to observe that KLF5 is markedly upregulated in human cholangiocarcinoma cells when compared with normal human cholangiocytes. So knowing now that KLF5 is upregulated in cholangiocarcinoma, we next decided to evaluate the role of KLF5 in cholangiocarcinoma pathogenesis. To do so, we first decided to genetic ablate KLF5 in cholangiocarcinoma cells using the CRISPR-Cas9 technology. We transfected cholangiocarcinoma cells with plasmids with the Cas9 and with guides to specifically delete the KLF5 gene. And after transfection, we sorted the cells with GFP. And after growing, we characterized these cells by RT-PCR, sequencing, and Western blot. We were able to generate 5 different clones with distinct genetic alterations. And when we measured the mRNA levels of KLF5 in all these clones, we observed that clone 2 and clone 20 displayed a marked reduction in the KLF5 mRNA expression when compared with CRISPR control cells. We corroborated this by measuring the protein levels of KLF5, and we also observed that the protein levels of KLF5 were completely decreased in clone 2 and clone 20 when compared with CRISPR control cells. We then move forward with the next analysis, selecting both these 2 clones. To evaluate the role of KLF5 depletion in CCA cells, we started by conducting a characterization by deep throughput deep proteomic analysis. And we evaluated the proteomic profile of CRISPR control cells, clone 2 and clone 20. We were able to identify more than 5,200 proteins, and in these volcano plots, we can see when comparing clone 2 with control cells or clone 2 with control cells, we were able to observe that proteins were upregulated, here marked in red, or some proteins were downregulated, here marked in blue. When we compare the proteomic profile of these 2 clones, the proteins that were differentially upregulated or downregulated, we came out with 417 proteins that were common between these 2 clones. And we then next decide to evaluate what is the role of these proteins that are commonly differentially regulated in the CRISPR KLF5 knockout cells. We conducted gene ontology analysis, and we observed that the proteins that were upregulated were mostly associated with distinct signaling pathways, associated with chemoresistance, with metabolism, with the regulation of cell cycle, and with the promotion of cell death. On the other way, the proteins that were downregulated were associated with migration and evasion, and also related with the regulation of oncogenic signaling pathways. So we know now that there are several signaling pathways that are altered in these KLF5 knockout cells, but do this results in functional alterations in these cells? So we next decide to evaluate cell growth and cell proliferation. When we cultured CRISPR control cells and both KLF5 knockout clones, we observed that while control cells grew very nicely over time, both clones displayed a decreased cell growth capacity when compared with control cells. And then when we confirmed these results by cell proliferation using flow cytometry, we were able to observe that both clone 2 and clone 20 displayed a marked reduction in cell proliferation, which was accompanied by a reduced expression in the typical proliferation-associated markers PCNA and Ki67. Associated with cell proliferation, we also measure cell cycle. And when we evaluated the distribution of the distinct cell cycle phases in clone 2 and clone 20 compared to control cells, we observed that both the KLF5 knockout cells displayed an cell cycle arrest at G1, that was further explained by a decreased expression in cyclin D1 and in CDC25A. Regarding colony formation, we observed also that the genetic ablation of KLF5 deeply impact on the colony formation capacity of AGI1 cells when compared with control cells. Regarding basal cell death, we measured apoptosis by flow cytometry, observing that at basal conditions, the CRISPR control cells die less when compared with both clone 2 and clone 20, which displayed increased cell death in basal conditions. Regarding migration, we, after conducting the scratch assay, we observed that the CRISPR control cells migrated more than the KLF5 knockout cells. And this was also true when we measured invasion and when measuring the expression levels of N-cadherin on PDGF1, PDGFA, two genes that are deeply involved with migration and invasion. These expression levels were markedly reduced after genetic ablation of KLF5 when compared with CRISPR control cells. So after realizing this phenotype in vitro, our next question was to how do these cells behave in vivo? So we transfected these cells with a luciferase expressing vector, and then we injected these cells subcutaneously in immunodeficient mice. We measured the growth over time, and we were very happy to observe that control cells grow over time, leading to the development of very nice tumors, while KLF5 knockout cells did not develop any tumor when implanted in immunodeficient mice. We then decided to implant these cells not in the subcutaneous flanks of these mice, but in the organ of origin of cholangiocarcinoma tumors, which is the liver. So we conducted an orthotopic model where we injected both CRISPR control cells and KLF5 knockout cells in the liver of immunodeficient mice. And we were very happy to observe that while mice implanted with CRISPR control cells, very nice tumors developed in the liver. No tumor was observed in the liver of mice injected with the CRISPR KLF5 knockout cells. Having these results with the genetic ablation of KLF5, we next question, can we target pharmacologically KLF5? So we took advantage of a potent selective KLF5 inhibitor, which is ML264. We treated normal human cholangiocytes and cholangiocarcinoma cell lines with distinct doses of this inhibitor from 0.25 to 25 micromolar, and we observed that while no differences were observed in normal human cholangiocytes, cell viability in cholangiocarcinoma cells was reduced in a dose-dependent manner in both of the cells. We confirmed these results by measuring cell proliferation by flow cytometry, and when incubated both AGI1 cells and HUCCT1 cells with 10 micromolar of ML264, cell proliferation was reduced after the treatment with the KLF5 inhibitor. So next we question, do this inhibitor also works in vivo? So we injected AGI1 cells subcutaneously in immunodeficient mice, and after randomization, we treated mice for 1 month with 5 milligrams kilogram of ML264 for 28 days. And we observed that when we started the treatment with ML264, tumor growth in vivo was completely abolished, while mice treated with vehicle displayed a marked growth in tumor growth. We also implanted AGI cells orthotopically in the liver of immunodeficient mice, and we treated these mice with a KLF5 inhibitor again. And in this case, we used cells that express luciferase in order to be able to monitor tumor growth over time. And we observed that while in mice treated with vehicle, we observed that tumor growth significantly increased from the beginning of the treatment until the sacrifice, while mice treated with the KLF5 inhibitor displayed no tumor growth when compared with control conditions. And here we can see, for example, some hematoxylin and leucine stainings that show that the tumor areas in control mice were far bigger than the mice treated with ML264, while when looking phenotypically to the liver, we observed very large tumors in the liver, while very small ones were observed in mice treated with the KLF5 inhibitor. So if you allow me to go back to the proteomic results, if you remember, there was one of the processes that were associated with KLF5 genetic ablation, which was chemoresistance. So our next step was to evaluate if KLF5 is also contributing to cholangiocarcinoma chemoresistance. And to do so, we used again the KLF5 knockout cells and treated these cells and the CRISPR-controlled ones with the current first-line chemotherapy, the combination of gemcitabine with cisplatin. And we measured both cell viability and cell death. And we were very happy to observe that GemCs-induced cell death was markedly increased in the KLF5 knockout cells when compared with CRISPR-controlled ones. And when measuring apoptosis by flow cytometry, we confirmed that GemCs-induced apoptosis was potentiated in the KLF5 knockout cells when compared with CRISPR-controlled cells. Finally, we decided to evaluate if we can combine this first-line chemotherapy, the combination of gemcitabine and cisplatin, with the KLF5 inhibitor to inhibit cholangiocarcinoma cell growth in vivo. To do so, firstly, we decided to evaluate in vitro if there is an addictive effect of combining GemCs with ML264 in reducing cell viability and inducing cell death. And we treated cells with the GemCs alone, with the KLF5 inhibitor alone, or with a combination of both. And we observed that while GemCs and ML264 reduced cell viability, this effect was more evident when we combined the chemotherapy with KLF5 inhibitor. When we measured apoptosis by flow cytometry, we were very surprised to observe that ML264 KLF5 inhibitor did not exert any apoptosis induction when used alone, but was able to potentiate GemCs apoptosis induction when treated CCA cells. Then we selected AGI1 cells to inject in mice now, subcutaneously, again in immunodeficient mice. And after randomization, we started treating mice with either vehicle control, with GemCs alone, with the KLF5 inhibitor alone, or then we combine both to see if we can abolish tumor growth. As you can see here in this graph, we were happy to observe that both the first-line chemotherapy and KLF5 inhibitor decreased tumor growth over time, but when we combined both, tumor growth was completely abolished after starting the treatment. So overall, in conclusion, we observed that KLF5 is upregulated in cholangiocarcinoma, contributed to cholangiocarcinogenesis by promoting cell survival and proliferation. We observed that both the genetic and pharmacological inhibition of KLF5 reduced cell proliferation, migration, invasion, and tumor growth in distinct experimental models of cholangiocarcinoma, also contributed to cholangiocarcinoma chemo resistance, and we hypothesized that the inhibition of KLF5 with ML264 may represent a potential therapeutic strategy for patients with cholangiocarcinoma, and this combination with the first-line chemotherapy might be of therapeutic benefit for patients with cholangiocarcinoma. So I'd like to thank our research group in Biodonostia, led by Professor Jesus Banales, Dr. Oyane Harris, which is a very talented researcher that started and was deeply involved in this research, our national and international collaborators, and last but not the least, I'd like to deeply thank AMMF for supporting this research, and thank you very much for your attention.